Hi friends, today we are going to see the past year NEET questions for the chapter Biotechnology Principles and Processes. Today in this video we are going to cover the last part of this chapter, which is part 3. Now let's begin with the first question here. Introduction of food plants developed by genetic engineering is not desirable because of dash. So it's moreover like an artificial method, right? They're not natural method. You're introducing of your own interest. So plant which are developed by genetic engineering are called as transgenic plants. Plants are genetically modified, which have genetically food that we consume. For their production, microorganisms like bacteria, virus, they are used as vectors to insert the required gene or to delete a particular gene sequence or whatever. So by consuming them, there is a danger of entry of virus or the toxins causing different types of allergies or other health hazards to the human beings. So we are taking something that is artificial. You are changing, modifying the system, the natural system, by the introduction of certain viruses or bacteria, which is used as vector, which carries a modified gene and modifies the whole set. Sometimes it might not be suitable for the people who consume it. So there is a danger of entry of viruses and toxins within the introduced group. So option D is the answer. Next question here. 2 bacteria found to be very useful in the genetic engineering experiments are dash. So one we know it is Escherichia, right, E. coli, and the other is Agrobacterium. Escherichia and Agrobacterium are both used as vectors in genetic engineering. While if you take Nitrobacter, Azotobacter, they convert the nitrites into nitrate. So they are basically nitrogen fixing bacteria. So they don't help in the transfer mechanism. Rhizobium diplococcus also helps in the same activity. They are symbiotic nitrogen fixing bacteria. So, and Nitromonas and Klebsiella are actually pathogenic bacteria. So they are also not used. Therefore, the correct answer to this question is option A. Next question here. The plasmids present in the bacterial cells are dash. Okay, again this is a very easy question for you. So we all know whenever we see a definition for plasmid, we always read it as a plasmid is an extra chromosomal double-stranded DNA. So the same thing will come here. So a plasmid which is present in the bacterial cells are circular double helical DNA molecules. Correct answer is option A. Next question here. The process of separation and purification of expressed protein before marketing is called as dash. Okay, you have various stages, right? So you have 4 options here: downstream bioprocessing, post-production processing, and upstream. So I will tell you what is upstream and downstream. Upstream processing refers to the first step in which the biomolecules are grown, usually by the bacterial or the mammalian cell lines in a bioreactor. So here if you see, we are talking about a process of separation and purification expressed protein and before marketing. So it's almost the last step that they are trying to convey here. It's the last previous step or something. So upstream is the beginning step. Obviously this cannot be the answer. So the overall process is simply called as bioprocessing. Again, this is not the answer. Post-production processing is after the completion of the process. Therefore, the correct answer to this question is downstream processing. Here if you see the various stages of processing that occur after the completion of fermentation or synthetic stage, they include separation and purification of the product, which is called as downstream processing. Therefore, the correct answer is option A. Next question here. In vitro clonal propagation in plants is characterized by dash, and this question was asked in the year 2014. Now, what do you mean by clonal propagation? Propagation is movement, right? What do you mean by clonal propagation? So when we talk about clonal propagation, it refers to the process of asexual reproduction by the multiplication of genetically identical copies of individual plants, or it can be used even for the other sectors. The term clone is used to represent a plant population that is derived from a single individual by the asexual type. So this is in terms of plants. Okay. Yeah, I'm talking about the plants only. But there is a method or there is a technique where you can characterize this. The technique is PCR and RAPD. Okay. So what is the full form of PCR? Polymerase. It's called as polymerase chain reaction. What is it called as? Polymerase. You should know this. Polymerase chain reaction. and the other step that you have, I mean the full form of RAPD, it's actually, I mean the full form is random amplification of polymorphic DNA. What is it called as? Random amplification of polymorphic DNA. It's very essential that you know the full forms of this. Sometimes you might get questions from this. Therefore, the correct answer to this question is option A. Next question here. Commonly used vectors for the genome sequencing is dash. Easy question, right? Human genome sequencing is a process that determines the complete DNA sequence of an organism. Okay. You determine the complete DNA sequence of an organism's genome at a single time. This requires sequencing of all organisms' chromosomal DNA as well as the DNA contained in the mitochondria. That is, for mammals or for animals it is one in mitochondria. For plants it will be in the chloroplast, right? Commonly used vectors for human genome is actually BAC. What is the full form of BAC? Bacterial artificial chromosome. Bacterial artificial chromosomes. And the other one is yeast artificial chromosomes. Okay, which is option B and C. Therefore, the correct answer is option B. Next question here. DNA fragments generated by the restriction endonucleases in a chemical reaction can be separated by dash. So this question was asked in the year 2013. So whenever we talk the process of separation, okay, you have a hint here, separation, and we already discussed in the part 1 and 2. Whenever we talk about a separation method, we use electrophoresis, which uses size and charge as the main criteria for the separation method. Now the same answer will go here. DNA fragments generated by the restriction endonuclease in a chemical reaction can be separated by a gel electrophoresis because DNA fragments will be negatively charged. So when you pass in current, it will be positive. So what happens? It moves towards the positive electrode. Okay. So the charged molecules will be separated by forcing them to move towards the anode under the electric field through the medium or a matrix. The DNA fragments separate according to their size and charge. Therefore, the correct answer is option B. Next question here. PCR and restriction fragment length polymorphism. So how do you abbreviate it as? RFLP. Okay. Are the methods of dash. What do they actually do? They actually help in genetic fingerprinting. So you have a diagram to your right side which shows a picture of DNA fingerprinting. You have a victim and during the crime scene you take in the fingerprints and you will have certain list of suspects and you can analyze to which it matches. This is a very good technology actually to identify the murderers, the suspects, the crime, the people who have committed crime and all that. Okay, therefore the correct answer is option D. Next question here. Which one is true statement regarding the DNA polymerase used in the PCR? Okay, the name of this DNA polymerase that we use is actually Taq polymerase. And where is it obtained from? It's a thermophilic bacteria which is called as Thermus aquaceus. That is the name of the bacteria. And I told you something unique about it. They are bacteria which can sustain or which can survive in high temperatures. That is why they are called as thermophilic. Okay. So now I think you'll be able to answer this. Yes, it remains active when at high temperatures. The name of this DNA polymerase is Taq polymerase, which is extracted from a thermophilic bacteria. And the main reason why do we use it? It's because they can withstand high temperatures. Therefore, the correct answer to this question is option D. Which is correct? Option D. Next question here. For transformation, microparticles coated with DNA to be bombarded with gene gun are made up of dash. So this is a direct question. For gene transfer into the host cell without using a vector. Okay. Where you don't use any vector, any source or any resource from outside, you will directly bombard it into the target cell. So for that you will use gold or tungsten. Okay, it is made up of tungsten and gold, which is coated with the foreign DNA, and it is bombarded into the target cell at a very high velocity. Therefore, the correct answer is gold or tungsten. We do not use silver, platinum, silicon or platinum, but we only use gold or tungsten. So it is coated with that. The foreign DNA will be coated with that, and when it is bombarded inside, when it is moved with high velocity, it will reach the target cell. Okay. Next question here. Biolistics gene gun is suitable for dash. Okay, biolistic is a direct gene transfer method for constructing what? Recombinant DNA. The gene gun was invented by John C. Stanford and by Edward Wolfe. A gene gun can be used to genetically infect the cell or the whole organism with the foreign DNA by aiming the barrel of the gun and firing it. So what does a gun do? It will fire the target. Likewise, when you coat it with gold and tungsten, along with that you have the foreign DNA. When you fire it, it will obviously go with high velocity where it will reach the target cell. So this is the main criteria that they use here. The micro-shot projectiles in this biolistic gene gun are made up of microscopic sized gold or platinum powders. It's actually gold or tungsten powders. These expensive powders are soaked in the DNA or RNA and are engineered for insertion into the genome of the cell. Therefore, the correct answer is for constructing recombinant DNA by joining two vectors. Okay, next question here. In genetic engineering, the antibiotics are used for dash. Why do we use antibiotics? Antibiotics, what is the natural work of an antibiotic? It works against infections and diseases, right? So antibiotics are powerful medicines that fight against bacterial infections. We all know that. They either kill the bacteria or keep them from reproducing. Further growth will be suppressed. In genetic engineering, the antibiotics are used as selective markers, selectable markers. So you can see the resistance there, how it is resistant or if it is immune to it, whatever. Okay, fine. Next question here. The figure below shows 3 steps A, B, C. Right? It shows A, B, C. It's a PCR. It's a polymerase chain reaction. Select the option given the correct identification together which it represents. Okay. I'll give you the answer here so that you can analyze now. The correct answer is C. Extension in the presence of heat stable DNA polymerase. So here if you see in option C, this particular C I'm talking about. So here you can see extension. So this is right. This represents the correct activity. Otherwise here it goes as a mistake. So here if you see, which statement out of the 4 are correct? If you see, correct is C, which they have correctly mentioned. It is an extension thing that is happening there and they have correctly mentioned there. Okay. Next question here. Agarose extracted from seaweeds finds uses in dash. Of course, the question is direct and I think all of you know the answer is yes, it is used in gel electrophoresis, where you prepare a gel with the help of agarose. It's a solidifying agent and it will not interfere with the reaction. In gel electrophoresis, agarose extracted from the seaweed are used in gel. Agarose made up of 0.7% of gel shows good resolution of large DNA and 2% of gel will show good resolution of small fragments. Okay. Next question here. What is true for a plasmid? Again, this is a very easy question. You have to analyze the options here. Plasmids are widely used in gene transfer. Yes, that is right. They are found in virus. Wrong. Plasmid contains gene for vital activities. Wrong. They are the main part of the chromosome. Wrong. The correct answer is they are widely used in gene transfer mechanism. The correct answer is A. In bacterial chromosomes, the nucleic acid polymers are dash. Again, this is an easy question for you because we just now discussed about it in the earlier parts. I told when you talk about a bacterial DNA, it is always circular. So here the option B is the answer. It's a circular DNA molecule, right? See, these questions have already been discussed a lot. I don't want to go repeatedly. So here by now you have guessed the answer. Yes, it is option B. Next question here. The plasmid is dash. Again, this is an easy question for you. We will analyze the options here. It helps in respiration. Wrong. Genes found inside the nucleus. Wrong. It is a component of the cell wall of the bacteria. Wrong. is the genetic part in addition to DNA in a microorganism? The option D is the right answer. So the term plasmid was introduced by Lederberg in 1952 for the extra genomic DNA segment. So it's a small circular DNA molecule that is found in the bacterial cell in addition to the large circular DNA. So you find it like you will have the bacterial DNA, along with that you also have this. That is how you can put it easily, right? Next question here. Organelle or organoid involved in the genetic engineering is dash. Again, the question is very easy for you. Yes, the answer is plasmid. So here you have a big description for it. So what does a plasmid do? It carries the DNA of the other organism known as cosmid, and that can be introduced into a desired cell. You do a genetic transfer, you call recombinant DNA. All that, if you see, we are introducing something from our interest. We are introducing something foreign into the natural product. So the organelle that is involved in genetic engineering is option A, plasmid. So with this we complete the full chapter, Biotechnology Principles and Processes. Kindly have a go through so that it will help you in your exams. Hope to meet you in a next new chapter. Thank you.